Hello, everyone, and thank you all for tuning in. I'm Nik Palevich, and today's talk is about applying a whole genome sequencing approach to improve our current diagnostics tools and understanding of Clostridia species that are associated with the spoilage of vacuum packaged meat. The work that I'm going to present today was largely all done pre-COVID-19 and was a great opportunity for some interdisciplinary collaboration with various members of the food and bio-based products team. Okay, so let's talk about genomics of Clostridium spoilage. First off, spoilage of chilled vacuum packaged meat by Clostridia species is commonly referred to as blown pack spoilage, or BPS for short. Blown pack spoilage is still a big challenge for the meat industry due to financial losses and reduced consumer confidence. And to the right there is a list of Clostridia species associated with spoilage, and in blue are those specifically linked to BPS. In particular, Clostridium estheticum and Clostridium estheticum subspecies laramiense, both linked to BPS around the world, especially the UK, America, New Zealand, and Ireland. Despite the importance of these spoilage microorganisms, studies on Clostridium estheticum are challenged by numerous aspects, and these include firstly, a lack or poor growth in laboratory media, long culturing periods, and unpredictable isolation on culture media. And this is largely due to the fact that there is no differential media or straightforward discriminatory methods available for the specific identification of different spoilage Clostridia species. Now, there are many factors that have been linked to the onset of BPS, those associated with the treatment of the meat during the food supply chain shown in blue, and also the various microbiological factors shown in green that are the focus of this project, such as the different Clostridia species, strain variability, and also their ability to form spores. These factors hamper the detection of Clostridium before occurrence of BPS, which further undermines efforts to prevent the occurrence of BPS. Now, nevertheless, considerable developments have taken place with regard to culture-independent methods, such as the development and use of PCR-based detection systems. However, the information on different Clostridia species and strains is limited and remains highly fragmented. To give an example of this, let's consider two scenarios. The first is an in vitro experiment in which a small amount of spores from the reference strain of Clostridium estheticum are inoculated onto beef, lamb, or venison vacuum packs and stored chilled. The pack will generally blow with associated proteolysis and offensive odor in around 6 weeks. Now let's look at the second example. If we consider the actual industry presentation based on meat stored chilled for up to 4 months, where we sometimes see blowing much later than that at around 10 weeks of storage. Here we have the bacterial 16S rRNA gene amplicon sequencing results from scenario 2. And these have been generated from DNA extracted directly using swabs from meat samples. Although we're looking at only a small sample population, what we can see is that Clostridia may be present throughout all stages of storage, and that some strains, for example, Clostridium 5 and 13, appear later in the biota. And these observations suggest that the current test may include species or strains that cause overt spoilage at different rates, some of which may not be significant commercially. Okay, so what could be the underlying issues with the PCR-based techniques? Well, there are three main technical aspects at play here, but the main take-home message here is that we get occurrence of false positives. So why whole genome sequencing? Currently, there is a very limited amount of knowledge of these economically important spoilage bacteria. Whole genome sequencing data provides the entire genetic code and metabolic potential at strain level resolution that will ultimately allow for the optimization of cultivation methods and exploration of the genetic characteristics of different Clostridia species. Ultimately, what we want to see is to improve on existing rapid and cost-effective pre-screening methods for, firstly, identifying different meat spoilage Clostridium species. Secondly, to determine spoilage potential of vacuum packaged meat. And lastly, to exclude the groups showing slow or no spoilage activity. With all that said, the main aim for this project was to isolate Clostridia species from spoiled meat samples and characterize these isolates using whole genome sequencing. Now, I understand that genomics is a bit of a niche topic among these talks, so I will focus more on the comparative analyses than on the nitty-gritty aspects of the genomes themselves. And this table summarizes all of the Clostridia genomes sequenced to date that are associated with BPS. Thus far, we have isolated, sequenced, and published 6 high-quality draft Clostridia genomes. For those wanting to know more or use these Clostridia genomes for their own future work, I've hyperlinked each isolate name and corresponding reference to their respective NCBI assembly and MRA publication. There is also a link to the bioproject for all the sequencing work at the bottom of the slide. Here are the 3 main comparative pangenomics aspects that will be presented in today's talk. First, we will look at how our Clostridia isolates align with other relevant Clostridia species at the whole genome level. We will then look at how our Clostridia isolates are similar or different to one another in terms of gene content. Then we will look at the carbohydrate-active enzymes or CAZyme profiles that shed light on the carbohydrate metabolism potential of our genomes. This tree represents the Clostridia pangenomes, which consists of 14 Clostridia. This is a functional genome distribution analysis that carries out an alignment of the entire genome sequences. The 14 genomes grouped into 2 distinct clusters and really point out the differences observed at the genome level, both between species groups and amongst individual strains or isolates. From our genomes, all but M14 grouped into cluster 1, with the Clostridium aestheticum and Clostridium tagloense type strains, with FP3 and FP4 as potentially new species. Next, we will investigate the cluster 1 genomes in a bit more detail to determine their unique gene complements that correlate with their growth characteristics that may separate them as discrete species. This Venn diagram illustrates another level to the pan and core genome comparisons, with the center representing the 292 gene core genome of these Clostridia isolates. The 5 genomes are colored into their respective species groups, with the outer numbers that are circled representing the total number of genes that are unique for that particular genome. As you can see, there are discrepancies both between the 5 species groups, but also within species groups and even amongst individual genomes too. So let's now talk about carbohydrate-active enzymes or CAZymes. The CAZymes have been around for about a decade now and extensively used in microbiome research. For those that want to know more, I've also hyperlinked each of the logos in this slide to the associated database web pages. And at the bottom of the slide, there are a couple of my recent works with particular focus on CAZy analysis. Briefly, here are the different groups of CAZymes, and each contains many different types of enzyme families that have very specific functional descriptions. The CAZy profile of a genome is a great place to start if you're wanting to know what kind of carbohydrates your organism is genetically able to utilize. And this information should always be followed up with actual experimental work to confirm such assumptions. Here we look at the CAZy profiles for each genome and the distribution of the 5 main CAZy families across the pangenome. The phylogenomic clustering to the left is based on the previously described FGD analysis and further corroborates the functional grouping of Clostridia into their respective clusters. As you can see, the CAZy profiles differ greatly between the different species, but not so much within the species groups. This indicates that indeed there are discrepancies in the substrate utilization preferences between different Clostridia species. To put these findings in context, here we look at the glycolytic pathway and other main pathways that strictly anaerobic bacteria use for energy production. For each of the different pathways, the presence or absence of each particular enzyme is noted with a colored designation to each of the individual genomes that are in the legend to the bottom right of the slide. As you can see, almost all the isolates possess the enzymatic machinery for the complete pathways for butyrate, formate, and acetate production, but some have much more complex metabolic capacities and are able to produce pyruvate through alternative methods or pathways, such as through lactate production via the methyl glyoxyl shunt or by utilizing uronic acids for growth. Lastly, in the previous financial year, we were able to extend this genomics work with a curiosity project that aimed to identify the nutrient and non-nutrient compounds that can be used to trigger Clostridium species spores to germinate outside their preferred environment for experimental or control purposes. To the right is a summary of the spore-related genes found in each of our Clostridia genomes. And to get these gene hits, a custom BLAST database was created for genes associated with spore germination using all publicly available Clostridia genomes. Then, with all our 6 genomes, were run through this custom database. I appreciate the font is a bit challenging, but I am hoping that these slides will be made available to everyone, and those really keen on the details of sporulation genes can look at the slide with a bit more detail. So what we have here is a interrogation of the gene cascades and metabolic pathways associated with spore germination compared across our 6 genomes. I've included the KEGG ontology or KO term designations for each gene as a reference. Now, the genes have been grouped into their biological categories, such as those associated with things like spore formation, receptors for spore germination. We have in green there the entire sporulation activation cascade pathway, also sporulation regulatory genes, etc., etc. The majority of genes from this analysis are universal for all our 6 genomes, but arguably the most important genes are listed in the blue box at the bottom and encode for proteins that respond to the presence or changes in concentration of certain ions and amino acids. Based on our results in current literature, it is apparent that various amino acids may act as possible cofactors or compounds that can induce but also strongly inhibit spore germination in meat spoilage-associated Clostridia. The value of this data is that it can be used to construct a putative model for nutrient and non-nutrient germination of Clostridia spores for downstream molecular and in vitro testing. Lastly, some conclusions and future work. Now, our genomes have all been sequenced using short-read MiSeq technology. However, as part of the recent Curiosity project, the DNA extractions for additional MinION or long-read sequencing have also been done for the purpose of generating improved assemblies that will be completed hopefully in the near future. And of course, acquiring more isolates for whole genome sequencing will facilitate more in-depth analyses and help us develop new tools for determination of Clostridia-associated spoilage potential. From the genomic data, we have a snapshot of the carbohydrate utilization and metabolic potential of these bacteria. But just because the DNA sequence tells us that these genes are present, it does not mean that they are indeed functional. So in order to get a true understanding of the substrates that can be used to improve on current cultivation methods, additional gene expression work needs to be done. And likewise, future RNA-seq work will greatly facilitate the discovery of genetic mechanisms associated with BPS and spore germination of these fascinating Clostridia species. Lastly, and for future work, we have shortlisted compounds and acquired a library of amino acids that should be further analyzed for their potential effects on induction or inhibition of spore germination at different points along the food supply chain. Thank you for tuning in.